Okay, hi everybody. This is LeClair. I am presenting to you for the first time online, so hopefully this is enjoyable. But today we'll be talking about a critique of Adams et al. 2019, which is a brief review of non-avian reptile environmental DNA, also called eDNA, with a case study of painted turtle under field conditions. So this paper is half review paper and half study, so we'll only be talking about half of it today. Now, things are obviously a little bit stressful and grim, so I'm going to give you a turtle trivia question, I suppose. So by the end, I'll give you the answer, but take a guess as to whether this painted turtle is a male or female. So I'll give you the answer at the end. So first, before we start anything, I want to make sure we know what eDNA is. So eDNA is this really cool technique that you can use to actually detect organisms without ever having to see them in the environment. And that's because we're constantly shedding DNA by our skin cells or feces or urine or other sources of DNA, and you can capture that in an environmental sample and actually extract that and amplify it in a lab and give you that positive identification of, yes, this organism was in the area of that sample that you took. So it's a really cool technique that allows us to find animals that are hard to find or even invasive species before they start invading a place early on. So not only can you actually detect the organisms you're looking for, but you can also quantify the amount of DNA you catch and try to relate that to the abundance of the organisms. So we can not only detect them, but also get an estimate as to how many animals were in that area. So it's a really cool technique, and it's something that I'm currently working on. So the objectives for the study were as follows. Number one, they wanted to determine if eDNA can be used to detect turtles. It's been tried a couple of times and a couple of studies have been successful. This one, not so much, and we'll talk about that in a little bit. And then they wanted to determine how the abundance of turtles relates to the amount of eDNA they capture, both of which tying into this overarching question, which is, can eDNA be used to detect and quantify threatened turtle populations? That's actually my thesis question. In case you've never seen what's called a qPCR read, so qPCR is this technique that we use to quantify the amount of DNA in a sample. This is what the readout looks like, and I did not know how to read this a few months ago, so if you struggle with this, that's totally fine. So along the bottom, you'll see cycle, and that's also called CQ value. So the sooner you start seeing a curve in a qPCR readout, that means that you probably have more DNA in your sample because it's constantly warming up and cooling down, warming up and cooling down, and that's this machine looking for the DNA in your sample. And if it detects it at an earlier cycle, that means you probably have more DNA. Whereas if it amplifies later, like these black, pink, or green lines showing up down here, that probably means that you have less. So if you see later on a low CQ value, that means that you have a lot more DNA, whereas something like around 40 would be a small amount of DNA. Their study design was pretty simple. They just had 4 man-made ponds that were all enclosed. They were equal size as well, so they were enclosed with an electric fence, I believe they said, and that was just to prevent the turtles from leaving the pond as well as other turtles from coming in. They wanted a known amount of turtles in each pond. So the first pond had no turtles. The second one had a low number, so that was 11. The third had a medium number, which was 23, and the fourth had a high number, which was 38 turtles. They took 250 milliliters of water every 3 days from a random point in the pond for 3 months and 4 ponds. So that was a total of 120 samples that they took. Now, an important thing when doing eDNA is having both a positive and a negative control, which they had. The positive control tells you that you are doing your technique right. So in this case, it was a blood sample and also aquarium water. So that should amplify in the lab. If it doesn't amplify, you have a problem with your technique. Then they also had negative controls, which is just distilled water. And if that amplifies, that tells you you have an issue with your technique because it should not amplify. So they filtered and amplified their samples in lab settings, and their hypothesis was as follows. So they more explicitly stated their HA here, so I'll read that first. It was the quantity of eDNA is positively related with quantity of turtles. So the more turtles you have, the more eDNA you should be collecting. So it's like this positive linear relationship is what they're saying. So the HO then would be inferred as quantity of eDNA is not positively related with quantity of turtles. So there's no significant relationship there. So not only that, but they hypothesized that there was a strict order of eDNA concentration from high to low in the following containers and areas. So they said the blood sample would have the most DNA, then the aquarium water, then the high density pond, medium density, low density, no turtle pond, and the negative control should have no DNA. They ran some pretty simple stats. So they ran each sample in triplicate and actually only ran these stats on each sample. So they didn't run any of these stats on the main population of samples. So they ran a standard deviation, which, if you need a reminder, is just a sort of measure of center of how the data is grouped or spread throughout the data set. And then same with the mean, also a measure of center. Standard error just gives you an idea of how much error to expect in your statistical calculations. So they just calculated those three for their samples that they ran in triplicate. Now they ran another statistic, and it's not the Kruskal-Wallis, but I do need to tell you what the Kruskal-Wallis is first to tell you what this other statistic is. So if you've never heard of it, the Kruskal-Wallis is also called this one-way ANOVA, which basically tells you, are these samples significantly different from each other, or are they all from the same population? So what they actually used was called a Jonck-Heers test, which is a variation of the Kruskal-Wallis. And it not only tests whether they're significantly different from each other, but tests if they are in a specific order. So remember, they were trying to determine if the eDNA concentration ranged from high to low based on the sample locations that they predicted. So revisiting the hypothesis, this is what their HO would look like, which is all the means would be equal to each other, whereas their HA would be the mean of their first container would be less than or equal to the second one, less than or equal to the third, and so on and so forth until they go through all of them. So let's talk about the results. Only one sample was considered significantly different from background variation, which is crazy. So that CQ value is 31.06, which is also not exactly low, considering that they had small ponds with no number, a known number of turtles. So to give you an idea of how that fits in with the data here, their negative controls had an average CQ of 40, with a pretty tight standard deviation and error, which is good. Same actually with the standard deviation and error for the positive controls. So it was really just right smack in the middle there, but their negative controls, and we're going to talk about this in a little bit, should not have even had a CQ value. So that's a little concerning. However, their Jonck-Heers had a strong p-value, it was less than 0.001, and it fit their prediction really well. So there's clearly this relationship of the quantity of DNA and the number of turtles in a location. So that's what this figure here is showing you. So you can see I'll see if I can draw this here. The tissue control, which was the blood, had the lowest CQ value as expected, and then the aquarium water, and then you get your high density pond, medium, and then I don't have to circle the rest here, but then it goes the low density, zero density, and then the negative control. So the low density and zero density, again, should not be even on this, but there they are. So they get to accept their HA and reject the HO, which tells them that eDNA is indeed positively related to turtle abundance. So let's go ahead and critique this paper, and I do have some critiques for this indeed. So the negative controls apparently were taken haphazardly, which who knows what that means. They're supposed to be taken at every step during the eDNA process to tell you where contamination is coming through. So who knows where it started, because clearly there was an issue with contamination because their negative controls were amplifying and they should not have. So it could have been contamination. It could have also been what's called non-target detection, which we'll talk about in just a second. But another really concerning thing here, which might relate to why they're getting so many positive hits on negative controls, was that apparently they didn't test the water that was being put into these ponds. So that would be essential for an eDNA, especially a controlled eDNA study, to determine whether or not turtle DNA was already present. Present. So that was pretty concerning. Another issue here, and this gets to the non-target amplification, their primers and probe, which are these reagents that are used to find and amplify the DNA in the lab, did not only amplify painted turtles in their study. It amplified other species as well. So if there are other turtles in the area or in the water that's being fed into the pond, that would give them positive results, which of course would be a false positive. So they could be getting the wrong information there. And this also was really um, not so much concerning, but just an opportunity lost in my mind. They only tested 28 of the 120 samples that they had. So that they said this was because of time and budget constraints, which is totally understandable. But if they took 120 samples, I really feel like they should have made an effort to sample more of them. I mean, they were less than 30 samples, which is what everybody kind of uses as the rule of thumb for minimal number of samples. But it's a bummer that they didn't amplify more. Now they also had this huge type 2 error window because their decision on whether or not a sample was significantly different from a negative control was based on if it was more than 7 CQ values from the negative control. That does not seem like it was calculated by them. It seems like it was pulled from another paper. So who knows if there were more samples that were actually statistically significant from each other. I have a feeling there would be because taking a look at the error bars on the previous graph, it looks like there should be some that were significantly different from zero. So, or not necessarily zero, but what should have been zero, which was the negative controls. So for that reason, that's why only one of them was considered not negative. Now, what kind of surprised me is that they didn't do a correlation or linear modeling of any sort, which I think would have actually been more useful to know than the Jonquiers test. It's good to know that there is a relationship with eDNA amount and turtle abundance, but having the standard curve, I think, is more important for the conservationist perspective, where you can kind of examine the amount of DNA you captured and try to compare that to the amount of turtles that might be in your sample area. So my guess is that would have been a lot more useful, and that's actually what I'm intending to use in my research. And then also actually testing for significance between the negative controls and the other samples, I think would have been ideal because again, that 7CQ I don't think was actually calculated. I think that was just pulled from another paper. So I think they should have tried a t-test for two means or try to run an ANOVA and just see how that turned out. So overall, they need to make some major improvements with that study design. There are just some big errors there. Their statistical analyses could have been a little bit stronger too, and that type 2 error, I think, really prevented the study from being as good as it could be. So that's the paper, and let's get to that trivia question here. So this is a male turtle, and you can tell because he has such long fingernails there. The male painted turtle will use his fingernails to like flutter in front of the female's face like this, which apparently is quite exciting to females. So they look kind of like Wolverine is how I heard somebody describe it once. So really cool species. So thanks for listening. Here's the full citation, and stay healthy, stay entertained, and I'll hopefully see you all soon.